This is Divakar Patil, and I'm gonna teach you today about the methods of microbial limit test. In previous lecture, we have learned about the how samples can be prepared for the microbial testing or microbial limit test, and different method which can be used for the treatment or microbial limit test for detecting the microbes present in the specific formula for specific sample. Okay, so today we're gonna study about the first method, that is microbial filtration method, sorry, membrane filtration method. Okay, in this method we have to prepare two samples. Okay, in this method we have to prepare two samples, each sample containing 10 ml of solutions. Okay, okay, each sample containing 10 ml of solution. Then we'll subject this sample to the filtration. Okay, whatever sample we having, or we want to filter that we that separate or filter out. Okay, that we will be separate with the help of filtration. Okay, in previous lecture I have shown you the image of what kind of filtration membrane filter we use. Okay, I am again showing you this picture that see, this kind of assembly for the filtration is used in membrane filtration method. Okay, here the membrane filter is placed. Okay, this is the one metal unit. Okay, okay, this is the mesh. This is the container in which the filtration or the the solution which is to be filtered is collected. Okay, this is the membrane filter. Okay, membrane filter which we can incubate according to required time. Okay, to see the microbial growth. Okay, then okay, we perform the filtration. Okay, the filter media that is used in filtration. Okay, the filter media that is used in this filtration can be uh, should have, not can be, should have porosity of 0.4 micrometer or micron. 0.4 micron means the mesh size or porosity, means the pore size should be 0.4 micron, means particle size of 0.4 or less can pass through that, not the particles which having the size more than 0.5 micron, and more than that, the 1 micron, 2 micron, 0.7, 8, 9, whatever. Okay, the particles having size less than 0.4 micron can only pass through this membrane. Okay, the particles which having size more than 0.4 cannot pass the membrane. Okay, after filtration we'll take out the filtrant. Okay, after filtration, take out membrane filter from the filtration assembly and subject them for washing by using a appropriate solvent. Means after filtration, okay, after filtration means what? See this image. After filtration means, after filtration, during filtration we'll pass the solution from this thing. Okay, we'll pass the solution from this mouth, and then it will go through this mesh, and then with the filtrant will be collected. Okay, after filtration means we'll separate these both things. Okay, these both joints. Okay, we'll separate these things, metal joints. Okay, and then we'll get the membrane which is been used for filtration purpose. Okay, okay, after this filtration membrane or filter membrane, okay, is then subjected to the specific solvent for getting the for sorry, not getting the, for washing the that specific filtration filtration assembly. Okay, then each membrane should be washed at least 3 times. Means we have to wash the filter membrane which is been used for the filtration, membrane filtration should be washed at least 3 times. Okay, or for three times with the with the specified solvent, whatever solvent we require, water or any organic solvent which is being used, or which is used. Okay, should be used in should be used to wash the membrane filter for 3 times. After washing the membrane, after washing the first membrane filter, okay, it is been placed in soybean casein digest media. Okay, it is been placed in soybean casein digest media, and after that the second part will be placed in pd agar media. Okay, pd agar media means potato dextrose agar media. Okay, or potato dextrose agar media. Okay, then these two medias will be placed in a petri dish, and in this petri dish will add the parts, those both parts of the membrane filter. Okay, one part will be added in soybean casein digest media, and another part will be added in pd agar media or gp agar media. Okay, now uh, okay, in last lecture I have told you that why pd agar media, sorry, why soybean casein media is used. Okay, let me show you another time. Okay, okay, see here, the soybean casein media is used for cultivation or growth of the aerobic microorganisms or fungi. Means in this method, okay, in this method we are using the soybean casein media. Okay, to check out that in that specified field sample is there any presence of fungus or aerobic bacteria is there. Okay, for that purpose we are using this agar media. Okay, sorry, not agar media, soybean casein media. Okay, next. Okay, subject the plate of soybean casein for incubation at temperature of 30 to 35 degrees Celsius for a period of 5 days. Means we have to incubate the plate of soybean casein media here. We have prepared two parts, in which two parts of membrane, where first part is added into the soybean casein and second part is added into the pd agar media. So here the soybean casein media is incubated, or the plate which is containing soybean casein media, which is incubated at 30 to 35 degrees Celsius for the for five days. Okay, and then second media plate, okay, second petri plate which is containing pd agar media is incubated at 20 to 25 degrees Celsius for 5 days. Okay, both plates are incubated for 5 days, but both are having different atmospheric condition or different temperature conditions. Okay, so finally after the incubation, determination and the total number of microbial colonies found on petri plate, and may make a use of this count in incubation calculation part to determine the total viable count of microorganism. Means after incubation, means after that 5 days, okay, after that incubation period, we have to count the microbial colonies present on the petri plate. Okay, we have what we have done, we have incubated that uh, medias, nutrient medias, okay, which having samples or which having the parts of filter membranes, okay, which is having sample. Okay, if there is any presence of microorganism, okay, if there is presence of any microorganism, then the presence of microorganisms on the uh, petri plate. Okay, if the sample contains microorganism or it is contaminated, it will contain the microorganisms. Okay, so these colonies of microorganisms are calculated, okay, are counted and then used for counting as a calculation part of counting that how many viable microorganisms are there in the specific sample. Okay, the total viable count for determining the total viable count of microorganisms. Okay, if the total viable count of microorganism is found within the pharmacopoeial limit, then the test is get passed, or if out of limit, then the test get fails. Okay, so after counting the amount of microorganisms present in the media, will count will check the pharmacopoeial limits for the specific samples. Okay, mentions in monographs, okay, specific limits of the samples, okay, that how much limit can be tolerate, okay, how much limit which can tolerate for this specific sample. If it is oil sample, it is a fatty sample, or whatever it may be, okay, it is semi solid solid sample or insoluble solid samples, whatever sample it may be, okay, we'll check the pharmacopoeial limit for that. And if that substance or that particular thing contains particular sample contains microorganisms in within limit, okay, then that can be said, or that batch or that sample can be said as it passed the test. Okay, but if the sample does not contains more than pharmacopoeial limit, the amount of microorganism present in that specific sample is more than pharmacopoeial limit, then we'll say that the specific sample is failed in passing the test or failed. Okay, so means the summary of this method is, okay, the summary of this method is, we have to prepare two samples. Okay, we have to prepare two samples. Okay, so next method is spread plate method. Okay, spread plate method. In this method, firstly prepare the two sterile petri plate for of agar media. In this, in previous method, we have used two different we have used soybean casein media, PG agar media. Same in this method, we also having two petri plates of different media. Okay, first plate is of soybean casein media, that is used for cultivation of aerobic bacteria or aerobic microorganisms, and second plate that contains PD agar media is for calculating the anaerobic bacteria or fungus. Okay, so in this we have to take 0.05 to 0.2 ml of sample solution. Okay, okay, this sample solution will be placed on the surface of the both petri plate. Okay, means what we have done in previous method, we have taken 10 ml of each sample and filtered through the membrane. Okay, in this method, we are going to take sample in between the range of 0.05 to 0.2 ml. Okay, these drops of the samples or this sample will be placed on the surface of agar media. Okay, separately means we have to place this system or place this solution separately on both the petri plates. Okay, then we'll spread this solution. Okay, we'll spread the sample solution on surface of agar media by using a glass spreader. Okay, with help of glass spreader or anything just like a petri plate, sorry, glass slide, which can be also used to spread the sample on the agar media. Okay, so we'll use the glass spreader to spread the sample on the on the surface of media. Okay, then finally after spreading, okay, after spreading the sample, okay, both the plates will be subjected for incubation. Okay, for the period of 5 days at a respective atmosphere. Okay, according to test or according to whatever temperature we want. Okay, according to that temperature we'll incubate that sample for 5 days. Okay, into the incubator. Okay, incubator is used to cultivate the samples, cultivate, or what we can say that to grow the to grow the microorganisms. Okay, to make the growth of microorganisms we use incubator, and in this incubator the samples or these media agar medias or nutrient media will be nutrient media will be placed. Okay, so nutrient media will be placed. Okay, then after 5 days of incubation we'll check the plates for microbial growth or microbial colonies formation. Okay, means what happens if there is any sample or sorry, in this sample if there is any microorganism present. Okay, whatever it may be, fungus, okay, anaerobic bacteria, then aerobic bacteria. Okay, whatever bacteria, sorry, microorganism is there is present in the uh, nutrient media, then there will be the growth of these particular microorganisms. Okay, if it contain microorganisms, after the specific incubation period, that is 5 days for this method. Okay, after this specific incubation period there will be the growth of microorganisms. Okay, there will be somewhat growth of microorganisms in the specific agar media. They will form the colonies, or there will be the group of the microorganism. Colonies of microorganism means the formation of group of microorganisms. Okay, and then after the formation or after the presence or occurring the colonies in the specific media. Okay, we'll go for the counting of the numbers of colonies generated. Okay, we'll go for the counting of number of colonies generated, means after the development of microbial colonies. Okay, we'll count the specific number of colonies generated, how many colonies are there. Okay, that will be helpful in final calculation. That will be helpful in final calculation of total viable count of microorganisms. Okay, and finally make a use of this count in calculation part to determine the viable total viable count of the microorganism. See I have told you that after incubating this microorganism in cultural media. Okay, we'll calculate the number of microorganism present in that specific media. Okay, and then we'll calculate for the total viable count of microorganisms. These colonies will help you to find out how to find out the total viable microorganisms present in the specific uh, nutrient media. Okay, after that, if the total viable count of microorganism is found within the pharmacopoeial limit, then the test is get passed, or if out of limit, then test get fails. Okay, means if the number of total viable count present or number of total viable count occurred, okay, obtained in the specific solution, sorry, so not solution, specific sample. Okay, that sample which was incubated in incubator for 5 days. Okay, in that sample, if there is a presence of microorganisms within the pharmacopoeial limit. Okay, I have previously told you that each sample have specific limit for microorganisms. Okay, if you are cultivating the microorganisms, or if you are incubating that specific solution or sample, okay, you will have space, you will have a standard limit for that. Okay, that how much amount of microorganism can be present in that specific cultivated sample. Okay, if the sample lies between the pharmacopoeial range, then it is said to be it has passed the test. Okay, it has passed the test. Okay, but if that sample didn't complete the requirement of the limit, which is to be present in the sample. Okay, if that sample is not containing the required limit of microorganisms, or if it is not in the pharmacopoeial limit, okay, it is not in the pharmacopoeial limit, then it will fail the test. Okay, it will fail the test. I hope you may get both these methods. Okay, the first one was membrane filter method. Okay, in that method we have used the membrane filter, okay, for cultivation of the microorganisms. Okay, this method, okay, where we have used 10 ml of samples each. Okay, and this this formulation, or this sample solution is passed through the filter, and these filter papers are cultivated with the help of the specific agar medias. Okay, this then after incubation we'll be having numbers of colonies of microorganisms present in the sample. If there is presence of microorganism in the sample. Okay, and second method is spread plate method. Okay, in that we didn't use the membrane filter. Okay, we have directly applied the solutions of microorganisms or specific sample solution of specific sample on agar media. Okay, on agar media, and that we have cultivated for the specific period, that is 5 days. Okay, for 5 days, and then we'll be determining the number of present microorganisms on the specific or that both nutrient media, that is PD agar media, and second one is soybean casein media. Okay, these both methods are used to calculate the amount of, or to calculate the total viable count present in the specific nutrient media. Okay, in short, these methods are used to determine the number of microorganisms present in specific sample. Okay, so next part is preservation of pharmaceutical products by using the antimicrobial. Wait. Okay. Next part is uh, preservation of pharmaceutical product by using antimicrobial agent. Antimicrobial agents are simply said to be the agent which inhibits the growth of microorganisms. Okay, these are the agents or these are the chemicals or substance, pharmaceutical substance which can be used, okay, which can be used to inhibit the microorganisms. Okay, if any solution is there or any system is there which is containing the microorganisms, or there may be chances of growth, chances of having the growth of microorganisms, that can be inhibit with the help of antimicrobial agents. Okay, means this agent used to kill the microorganisms or reduce the activity or retard the activities of microorganisms. However, these antimicrobial agents are considered as one of the integral part of pharmaceutical pollution. Okay, these microbial agents are considered as one of the integral part of pharmaceutical formulation. Thus, these antimicrobial agents are basically added as preservative in formulation to preserve the formulation for long period of time during its storage condition. Okay, these are important part of pharmaceutical product, because if in any pharmaceutical product, if there is growth of microorganisms, or there may get growth of microorganism, if there is any growth of microorganism, then that product may fail the taste, or may be having less effectivity, or whatever effect it having, the formulation having, it will be kept affected by this product or these microbes present in that formulation. So this to kill this microorganism, okay, to prevent the pharmaceutical product from these microorganisms, we use this preservative as a preservative. Okay, what? The microbial agent. Okay, we use this microbial agent as a preservative, okay, to preserve the pharmaceutical product for longer period of time. Suppose we are having a syrup solution. Okay, so there is presence of sugar in the syrup. Okay, and sugar is a good agent for growth of microorganisms. Okay, so along with the sugar, or along with the API, or along with any other non-active pharmaceutical ingredient, we also have to add the preservatives for this formulation, okay, to prevent it from the microbial growth. Okay, and if there is any growth of microorganism, then it will spoil the pharmaceutical product. Okay, so we use these microbial antimicrobial agent as a preservative. Okay, hence the chemical additives which we are adding with an intention of prevention of pharmaceutical product belongs to the class of chemical preservative. Means the agents we use to prevent the microbial spoilage. Okay, the agent we use to prevent the microbial spoilage, or these are the chemical additives which are used to prevent the pharmaceutical product. Okay, which are used to prevent the pharmaceutical product are called as preservatives. Okay, these are chemical agent. Okay, which kills the microorganisms, or reduces the growth of microorganisms, or retards the action on the pharmaceutical product. Okay, if pharmaceutical product get affected by the microbes, then there will be the less effectivity of the specific formulation. Okay, less effectivity of the formulation, then efficacy, okay, stability, everything will be changed. Okay, everything will be changed. Okay, we also adopt the various physical method like sterilization, pasteurization, adjustment, okay, temperature adjustment, and low nutrient supply for for inhibiting and for controlling the growth of microorganism. Okay, so means preservative means not just adding the chemicals into the formulation. Okay, there are different kind of methods or different kind of things we adapt to control the growth of microorganisms. These methods are sterilization. We use sterilization for sterilizing the specific system instrument we use to avoid the microbial growth. Okay, then pasteurization, same thing, the pH adjustment. Okay, then the temperature adjustment. Okay, the low nutrient supply. Okay, for inhibition and control of growth of microorganisms. These are different methods, or these are techniques which can be used to control the growth of microorganisms. Okay, which will help us to make the pharmaceutical product free from microorganisms. Okay, okay, these are the methods which can be used, and also we use the chemical agents as a preservative for prevention of microorganisms. Okay, these chemical preservatives are basically get classified into four major groups, such as acidic, basic, mercurial, quaternary ammonium compounds. Okay, there are the four different types of preservatives. Okay, which are, as I told you now. Okay, then we'll see the examples of different different preservative which can be used as in different pharmaceutical products. Okay, so in tablet we use methyl paraben, phenol, cresol, benzyl alcohol. Okay, these are preservative. You may have heard name about you may have heard about these chemicals. Okay, which can be used in formulation of tablet. Okay, we use this as a preservative to avoid the growth of microorganism. In eye drops we use benzalkonium chloride, phenyl mercurial nitrate, chlorhexidine acetate. These are different chemicals which are used in eye drops preparation. Okay, because if there is presence of microorganism in eye drops, okay, it may affect the eyes. Okay, and as it affect the eyes, there will be problem in vision of patient. Okay, it may create problems with the vision. Okay, so we use this preservative as a for prevention of microorganisms. Okay, then in liquids or in mixtures we use alcohol, methyl paraben, chloroform, benzalkonium chloride. These are different chemicals which are used in the pharmaceutical product for prevention of that product from specific microorganism. Okay, avoid the growth of microorganism, to avoid the harmful effect which will get after the infection of microorganisms. Okay, to take care of the patient. Okay, if we didn't check about the sterility of this specific pharmaceutical product. Okay, and if that product is affected by the microorganism, then that will indirectly, or we can say directly, cause harmful effect on the body of patient. Okay, so for making it sure that making it sure, or for making the specific sample free from microbes, that specific formulation free from microbes, we use these preservatives. Okay, these are chemical preservatives which can be used in a pharmaceutical formulation. Okay, I hope you may have understand this topic, whatever I have told you today. Okay, so first topic was the methods of microbial limit test, in which we have first saw the test about the microbial filtration method. Okay, this test was about determination of microbes in specific sample. The same test then another method was spread plate method. This is also used to determine the amount of microorganism present in the specific sample. And lastly we have seen the how preservatives can be used in a pharmaceutical product. Okay, how that can be used to prevent the specific product from microbial agents. Okay, so in Next part will be studying about the ideal requirement, properties of antimicrobial agents. Okay, in tomorrow's lecture we'll see the ideal requirement and properties of antimicrobial agent. Okay, so I end this lecture with this topic.